Ready? Go.
Hi, I'm George Fadden, and this is my presentation. Antigentic variation in Trypanosome brucei. I'll probably mess up the pronunciation of that a million times during this, so bear with me. Picture of the trypanosome in the blue and red blood cells. Human African trypanosomes, also referred to as African sleeping sickness, only occurs in sub-Saharan Africa. It's caused by two different subspecies. One is the brucei gamians, and the other is the brucei rhodesians. Wild and domestic animals can host these parasites and may represent, under particular conditions, an important reservoir of infection for the tsetse flies. How is it transmitted? The sleeping sickness is most commonly spread through the bite of an infected tsetse fly. Even in the areas where the disease occurs, only a small percentage of flies are generally infected. Flies are a little bigger than an average housefly, about 1.5 times. They usually bite in the daytime, they are quite painful, and the blood meal or the bloodworm is how it's infected, or how it's transmitted. Here's a picture of a nice full tsetse fly. Obviously, bit a cattle or another human with the disease, and that can transfer to another person. Symptoms and causative agents. Starts with disrupted circadian rhythm, fever, headaches, muscle and joint aches. The late stages of it can lead to mental deterioration, coma, and death. Really important fact that with no treatment, it's 100% fatal. The problems. Vaccine would be a really easy way to eradicate it or at least bring the numbers down, but you can't use a vaccine with it. The problem is because the variant surface glycoproteins, or FSGs, they allow it to be undetected, or it's constantly changing its shell. So if you did have vaccine that would go towards one variation, it would change, and then there'd be no good anymore. So you'd have to make 1,500 different vaccines. They build up the resistance to the current ones, and it causes death to humans and cattle. How it evades the immune system? Many viruses, bacteria, parasites have developed different ways of avoiding the immune system. Each year we hit by strains of flu, viruses, they mutate, or with disinfectants. You have to use it for such a period of time, it no longer works. The African trypanosome affects our blood, organs, and tissues, but unlike viruses, they do not invade our cells. Therefore, they were continuously exposed to the antibodies, which we generate against them and circulate in our bloodstream. So as they multiply, we make antibodies against them. Around 5 to 7 days or a week, the antibodies destroy most of the trypanosome, and it decreases, but a new wave comes right back. They're unaffected by the antibodies against them because the immune system has to start over again. So there's no memory cells that are already infected to get a quicker attack on the foreign objects in the body. This process continues essentially indefinitely until the death of the host. During this time, we continue to make primary responses against new antigens. VSGs. First time they really found that this might have something to do with why they keep changing the forms. In 1979, the scientists by Keith Vickerman drew attention to a feature of the surface of an infected form of the trypanosome. Vickerman suggested that the surface coat might contain antigens responsible for the antigenic variation or change. The surface coat of every individual trypanosome was formed by 10 million copies of a singular molecular species of antigen. These antigens are also called glycoproteins because they consist of a protein backbone with sugar, glycoattachments. To switch the surface coat, the trypanosome can select any member of a large family of what we call the variant surface glycoproteins, or VSG family. A couple different options for switching through transcription, recombination, and allometric VSGs. How does trypanosome express only one of the hundreds of VSGs at one time in an individual cell? There's a couple ways to look at it. Two important questions arise when it comes to what regulates the timing and switching and the order that the genes are expressed. This kind of allelic choice is not unique just to the trypanosomes. For example, detection of odors in the nose of animals involves the use of a large family of odor receptors, but the sensory cells in the nose only express one member of this family, despite containing all the genes. There are other examples in animals of cells that are able to selectively express single genes from the families of the complexity of the various ones. The second question, or option 2, is that the VSG gene that is expressed is always located very close to the end of the chromosome, or the telomere. This is because about 20 telomeres contain VSGs, and that's necessary to configure for the expression of them. Trypanosome has 2 copies, or at least 11 chromosomes, hence they have at least 44 telomeres. In some respects, this reduces the complexity of the exclusiveness and questions why only one of 20 telomeric VSG genes are expressed. We know there are 2 main mechanisms by which VSG gene can be switched. One can switch the telomeric expression site that is being transcribed, or one can shuttle the VSG genes from other locations in the chromosome into an active expression site. For now, control is all we can really hope for, and as we learn more about this, maybe attacking the VSGs or finding out how the switching happens might be a better way to look at it, or a more treatable way to look at it. Continuing and improved case surveillance with screening of humans in at-risk areas and also domestic cattle can keep these numbers down for the time being. It requires political will and stability and funding, like anything, especially in third world countries. Better treatment of the disease and the animals, and to public awareness. It doesn't happen. There is an American form of trypanosome, not nearly as fatal as this, but if it's not happening in your own backyard, sometimes there's not as much attention and funding paid to it. This is my works cited, and that's it. Thanks.